Hello, and welcome to an abbreviated overview of the Central Dogma. DNA. It's the stuff of life, the information it needs to be able to carry out critical functions. But what good is information really if it can't be acted on? It's a lot harder than you might think to pick out some blueprints from the library when you're working without a consciousness.
If only there were some way to automate a system that can go from DNA to RNA to polypeptide, so that it's self-regulated, needs no intelligent manipulation. Oh wait!
We'll start with a prokaryotic cell, which is simpler. Now, let's talk transcription. Every good gene needs a starting point. One might think that the first codon of our final polypeptide will do. So where is it? Looks like things are going to have to be a little more foolproof. Let's pretend we're the cell and define a chunk of DNA to be the promoter, to make it easy to find. Then we'll make a widget that recognizes it, most notably the -35 and -10 boxes, so named for how far they are from the first transcribed nucleotide. This is called a sigma factor, and during initiation, it temporarily attaches to RNA polymerase, brings it over, and sends it on its way. There's something here too called the ribosomal binding site, or RBS, which is where the ribosome will go to bind. Inventive, I know. Some sigma factors will more actively bring RNA polymerase to their respective genes under certain conditions, but sigmas are always necessary for transcription to run smoothly. RNA polymerase will sometimes try to transcribe DNA on its own, but it'll almost always fall off before it does anything useful. During the elongation phase, RNA polymerase pulls apart the double-stranded DNA to expose the template strand, to which it then matches base pairs with hydrogen bonds. To build the mRNA strand, it attaches the adjacent phosphate group of NTPs to the OH group on the last 3' carbon, using the energy stored in the two phosphates on the tail end. That means that on this strand, the left is the 5' end, and the right is the 3' end. A few of these mRNA bases are hydrogen bonded to the DNA at any one time, keeping the whole complex relatively stable. Transcription is usually terminated in one of two ways. In row-independent termination, the transcript has a section that loops onto itself. This hairpin structure begins to form just as the RNA polymerase reaches a U-rich region, where it lengthens the mRNA with uracil bases. Since A and U only share 2 hydrogen bonds compared to the 3 of G and C, this makes it easier to tear the transcript away from the DNA and end transcription. In row-dependent termination, a row factor finds its way onto the transcript, then runs up to the RNA polymerase and takes it off the DNA.
Okay, here's a question now. Are the promoter, RBS, and termination sequence on the mRNA transcript? Take a few moments, think about it, write down your answers. All set? Here we go. The promoter is a sequence, recognized by sigma, that tells RNA polymerase where it should start, so it needs to come before the transcript. Remember, the +1 site comes after the -10 and -35 boxes. The termination sequence isn't always consistent, actually, and it can be longer or shorter, all depends on when the RNA polymerase finally decides to dissociate. The RBS, however, is and must be fully on the transcript, because that's what the ribosome looks for in order to start translation.
And on that note, here is a ribosome. Well, the small subunit, at least. For initiation, it finds that RBS. The Shine-Dalgarno sequence AGG-AGG is pretty common. It's a bit like the way sigma recognized the promoter earlier. Friendly reminder that this is the 5' end of the mRNA. It then slides down to the first methionine codon it can find, where the corresponding tRNA attaches, and then the large subunit falls into place. This becomes the start codon, and defines the open reading frame. That's how we know that these sets of 3, and not these or these, are the real codons for the polypeptide. During elongation, a new tRNA enters through a convenient channel up top into the A site. The P site is where the ribosome's peptidyltransferase activity occurs.
That's just connecting the chain to the next amino acid and then breaking it off the tRNA.
The whole ribosome also shifts over, but the tRNAs stay in place, so that they now occupy different sites. This is called translocation, and its direction is from 5' to 3'. The empty A site can now accept another tRNA, and that's all there is to it. For each amino acid, this is the N end, and this is the C end, so the polypeptide grows N to C as it comes out the top. Now, if a wrong tRNA comes in and imperfectly binds to the codon, the ribosome stays in place until the right one makes its way in. Don't worry, nature's thought of everything. It's important to note that the codon, and not the amino acid per se, is the target of this specificity. If a mutation or a particularly crafty scientist swaps out one amino acid for another on the tRNA that matches the codon, the ribosome would be none the wiser. Empty tRNAs are recharged by enzymes known as aminoacyl tRNA synthetase. They have a special site to accept the tRNA, its corresponding amino acid, and an ATP molecule. And with a little bit of magic, ta-da, good as new. Each tRNA has its own specific synthetase where it goes to be charged, determined by the variations in shape of the particular enzyme, tRNA, and amino acid combination. Translation terminates with the stop codon in the reading frame. Obviously, an artificial stop spanning 2 codons doesn't actually count. Just like this is not an actual methionine codon. Notice that translation does not start at the first nucleotide of the mRNA, nor does it stop at the last. These sections are descriptively called the 5' and 3' untranslated regions. Let's step back and have another look at all this. Transcribing DNA to mRNA requires a promoter and termination sequence. The transcript itself has a 5' and 3' UTR, and the former contains an RBS.
The polypeptide is coded by a sequence, the ORF, between the start and stop codons.
So we've seen now that one gene contains more information than what is actually transcribed and what is actually translated. At each step along the way, some nucleotides don't make it to the next step.
Therefore, not all base pairs in the DNA have identical rules. But does that mean they're less important? Well, you tell me. Question number 2. Will we get an mRNA transcript, or a protein, if the promoter is mutated? The RBS? The start codon? Again, write something down. It's fun, I promise. This might be easiest if we had a table to fill in. With a mutated promoter, there will be no transcript and no protein. If only the RBS has a problem, transcription occurs just fine, but translation doesn't start, so no protein. If the start codon disappears, then we still get a protein. There's got to be 3 bases spelling out AUG somewhere. But it won't be the right one. It could even be out of frame, which ruins everything. There's a chance it could still be salvaged if the next AUG maintained the same reading frame, and was so close to the start that no functional regions were lost. Well, I think that about covers it. Nothing else really comes to mind that we have to... there's... eukaryotes. Okay, eukaryotes. To find the DNA, we must be in the nucleus, where there are no ribosomes, so the translation will come later. And eukaryotes have no sigma factors to attach to the promoter. They have basal transcription factors instead, which are similar. There's just a lot more of them. Further upstream are proximal promoter elements, for additional regulation. And now, let's meet the activator, whose job is to bind to an enhancer, attach, or mediator, to the promoter complex, and increase the odds of starting transcription. Similarly, a repressor binds to a silencer sequence and decreases the transcription rate. These things are generally not optional. Their combined efforts are necessary to ensure gene transcription at suitable levels. Notice the introns. They're not remarkable now, but they will be soon. For termination, instead of rho factors, we have two elaborately named types of proteins at work:
cleavage stimulating factor and cleavage polyadenylation specificity factor. They do what their names imply and cut the mRNA at a recognized sequence. The RNA polymerase will keep transcribing for a few nucleotides until it comes off on its own.
Now for some processing. A 5' methylguanine cap goes onto the front. Poly-A polymerase adds a tail of many adenines. Spliceosomes recognize characteristic sequences of introns and cut them out. And now some proteins escort it to a pore in the nuclear membrane, where the mRNA finally crosses to the cytoplasm to be translated. Phew. If you're wondering about the greater significance of all this, consider that in any form of life, anything that isn't itself a protein is made by a protein. Yes, even the proteins that make other proteins. Wasn't always this way. We think the first life forms used RNA to handle their catalytic needs. In fact, the peptidyl transferase activity in present-day ribosomes comes from not the proteins, but the nucleic acids. And then amino acids, with their much greater variety and versatility, took over in dominant fashion. Proteins rule the world now. And let's be real, even our metal, plastic, and electronic devices are built by machines, built by humans, built ultimately by proteins. Nearly everything around us starts off with the assembly of polypeptides from DNA information. So maybe there's a reason why we call this the central dogma of biology.